Okay, so for some background information, alpha-synuclein plays an important role in Parkinson's disease, PD. It is the second most common neurodegenerative disease in the United States, and it is characterized by the presence of Lewy bodies in the substantia nigra, which is a part of the brain. So early symptoms include movement difficulties, and as the disease progresses, it can lead to a loss of speech and can potentially be lethal. Synuclein is a main constituent in these Lewy bodies, and phosphorylation at serine 129, an amino acid in the protein chain, has been reported to enhance its toxicity and is critical to the pathology of PD. So we are researching this because the mechanism of synuclein spread between cells is not fully understood. For our objectives, we have two smaller objectives, which are determining if synuclein can be phosphorylated in vitro or outside of a living organism, and determining if synuclein fibrils can be uptaken into differentiated neurons. And for our cells, we used neuroblastoma cells, which is a cancer cell line, and we wanted to differentiate them so they would be more neuron-like. And our primary objective was quantifying the difference in uptake across the different forms of synuclein. So non-phosphorylated synuclein, monomer and fibril, and then their phosphorylated counterparts so 4 samples in total. And after growing the cells and treating them with the samples, we did cell staining and fixation. So we grew the cells on a small cover slip. And then after reaching desired confluence, we created a cell mass stain for the cells so it would stain the cell membrane and then submerge them in paraformaldehyde so it would fix them in one state. So let's say it was in the middle of apoptosis, we would see it in that state in that moment. And then we could use it for fluorescence microscopy. And then here are Western blot results, which we just did to make sure that the proteins were present in our 4 samples. So we did monomer, phosphorylated monomer, fibril, and phosphorylated fibril. And on the left, you can see that we just tested for synuclein. So all 4 samples had synuclein. And then on the right, we tested for phosphorylated synuclein. And it makes sense that it's only in the phosphorylated fibril and the phosphorylated monomer. And then more importantly is the fluorescence microscopy results. In the green, it is the synuclein uptake. Here's the monomer, and the red is the cell mass stain. So it makes sure when we merge it that the monomer or the fibril that we're seeing is actually within the cell. It's not just extracellular. And then this little white bar at the right is 10 micrometers, just to see how big the cell is. Here you can see that the monomer is a pretty light signal. You can still see some green, but it's not very opaque. And then we move on to the phosphorylated monomer, and it's a little bit more bright, some more signal here in the middle, as you can see by the brighter green. And then again, the red just to make sure that it's within the cell. And then we move on to fibril, and you can see the uptake is even stronger by this brighter green on the left, this little aggregate. And again, making sure it's within the cell, not extracellular. And then most importantly is the phosphorylated synuclein fibril, and you can see there's a lot more. This is how most of the cells look, because we looked at 20 cells for each, and most of them had this amount of aggregates, and it was this type of bright green, opaque, and then again, making sure that it's within the cell. So for the conclusion, we hit our objectives. The in vitro phosphorylation confirmed that the fibrils can be phosphorylated, and then the cell mass stain allowed us to ensure that the synuclein was inside of a cell, so it did uptake into the differentiated neurons. And then for our next steps, hopefully we can identify a protein on the nerve cell's membrane that act as a receptor for phosphorylated synuclein aggregates. And then acknowledgements, thank you to everybody at Sheckman Lab, especially my mentor, Shenjie Wu, my PI, Randy Sheckman, and then Nathan Levy, an undergrad who taught me how to navigate the lab. And then obviously everybody at SSRP, Ellen, David, Roy, Lisa, and then shout out to the organization, CERM, who funded my project.